and it is a great pleasure for me to have a talk or a virtual platform. And few days back, Madam, she called me and she invited me for giving a talk. And I took an opportunity. One small correction: I'm retired now. Last year, I retired from my university job, but still I'm working as a visiting faculty with the same university, with the same department. So it is a indeed pleasure for me to have all distinguished listeners, distinguished people attending this virtual meeting. Rama Madam, Ashok Sir, Jaishree Madam, and all other respected faculty members. So I'm very much thankful to all of you for inviting me. As Madam suggested, that today's my topic is biocrystals in human health and in vitro studies with medicinal plants. On the left-hand side is a building of our university, right-hand side where I worked for nearly 40 years. Now, biomaterial crystals, the two opposite things are shown over here. There are two opposite things. For industrial purpose, we require large crystals, good quality, and perfection. And for biomaterials, the opposite things are required. We don't want that crystals to be grown in our body, and we want them, if at all they are grown, they should be in a very small size. So the two opposite things are there. For industry purpose, we require large size, perfections, and large quantities. In our human body, we don't want them to be grown in our body. So that should be no growth of crystals, ideally speaking. And if at all they are growing, it must be very small in size. Now, occurrence of biocrystals, there are three broad areas. One, they are occurring in human, then plants, and animals. The crystal growth of specific biomaterial compounds occur in body or living organisms. There are many reasons, and various pathogenesis and theories are proposed. So biocrystals encompass various crystals responsible for different ailments, human suffering, and health-related problems. Now, biomineralization is a very important process taking place in biocrystallization. And the factors which are influencing, they are categorized over here. And the environment is very important. Then biological, biochemical, and physiochemical. And then again, controlled mineralization and induced mineralization. So these factors are very important. In other category, the inorganic physicochemical process, and where the geological minerals are being deposited in human beings. So these are the broad categories where the biomineralization is taking place. Now, main factors, they are influencing the biocrystals. So main focus of the study of biocrystals, the first is how we study the biocrystals. So the one important part is the growth phenomenon of biocrystals. So how they are growing in body, and to study the growth of crystals in human being, it's a very important thing. Another important thing is the parameters affecting the growth of biocrystals. So various parameters are affecting on this growth, and it is very important to study, because by studying these parameters, we can control their growth. Then morphology of biocrystals, morphology or shape is very important thing. Suppose the crystals are spiky, then when they move in a vessel, they scratch and the bleeding is taking place. But if they are smooth, then no bleeding or comparatively less harm is taking place. Then in vitro studies are very important. We are focusing on that in vitro studies because we are trying to mimic the atmosphere occurring in a human body. Then mechanism of growth and dissolution. Again, it is an area of our interest. First, we have to study the mechanism of growth of biocrystals, and then we have to focus that how they are dissolved. So these two things are very important, and growth inhibition and dissolution of biocrystals. So growth is taking place, and we have to see the conditions, under which condition that dissolution is taking place. So dissolution of crystals under various conditions is very important to study. Characterization of biocrystals. Whenever we have any biocrystal growing in our body, whenever it is removed, the characterization is very important. And there are several characterization techniques available, like powder X-ray diffraction technique, then Fourier transform, infrared spectroscopic study, then various other spectroscopic study, then thermogravimetry, and various type of analysis available to characterize the biocrystals. The types of biocrystals in a living body. The biocrystals, they have three broad categories. One is organic, another is inorganic, and mixture of the both, organic and inorganic both. Then biocrystals of spatial interest. So we are going to focus only on three different type of biocrystals in today's talk, because today's talk is quite elementary one. And it covers the basic things regarding the biocrystals occurring and how to dissolve them or how to inhibit their growth. So we are going to focus on that. And in my next talk, I will go into the much more technical details and our results. So today is some sort of an introductory talk. So we are going to focus only on the three main aspects of biocrystals. One is urolithiasis, then atheropathies and gallstones and atheromatous plaques. Why to carry out studies on biocrystals? So these elements can be dealt with considering three main aspects. One is the prevention, then relief and cure. All three things can be dealt by our study. And one is growth is of crystals. That relief is something like if the growth is reduced or inhibited, then the relief is done. And cure, then once the crystals are grown by some mechanism, they are dissolved completely, then cure is taking place. So all the three different aspects can be dealt with the present study. Now we are coming to different type of biomaterial crystals. So biomaterial crystals are very beautiful. They look very beautiful, but they are painful and they are not very comfortable for the patients. So biomaterials or biomineral crystals induce several diseases in human body, even in animals also. And protein and crystals are grown. And this is the photograph of a protein crystal. So protein crystal and virus crystals can be grown. Then urinary stone problem. We have shown the cartoon. In a top cartoon picture, the healthy kidney, so the face is laughing, and the kidney with a lot of crystals and lot of stone. So kidney is in a sad phase. So urinary stone occurs in a body, which is very painful. And urinary stones are crystalline depositions. Then occurrence of urinary crystals in world. So 12% population of European Union is expected to be suffering from kidney stone problem. And 15% population of North India are again expected to be suffering from urinary stone problem. And there are stone belts, particularly in our region, Saurashtra. There are many places where people are suffering from urinary stone problems. So they are known as stone belts. These are the chemical constituents of different type of crystals, which are composed of the calcium oxalate monohydrate, the calcium oxalate dihydrate, calcium hydrogen phosphate dihydrate, tricalcium phosphate, magnesium, ammonium, phosphate hexahydrate. So these are the crystals, colite and struvite, and we have grown most of these crystals in our laboratory. So these crystals we have grown in our laboratory. These are some humor expected or associated with kidney stone problem. So in one cartoon with a chisel and hammer, the doctor is trying to remove the kidney stone from a patient. And in other cartoon, the kidney stone itself is giving its experience to small kidney stones. I'm sorry, kids, but last night your father passed him out. Father is not passed away, but passed him out. And another thing is in a Marathi, the person feels that the stone is something like a stone required for kapchi, for paving the road, but it is not that. It is something like a stone from the urinary tract. So Nasik stone and urological center, the common place in this cartoon. So these oxalate crystals are one of the main crystals occurring in urinary stone. And they are also known as a mulberry stone, covered with sharp projectiles or projections. And they mask, whenever they pass, they bleed things, and they are very hard and they are radio opaque. So these are the photograph of the calcium oxalate stone, as well as how they look in a urine, that is shown in the next slide. The phosphate stones are usually called as a calcium phosphate, and calcium, magnesium, ammonium phosphate or triple phosphate. And sometimes they look like a dirty white, and they are also radio opaque. And calcium phosphate is also known as a bruchite, and they also appear like a needle under microscope. So the photograph is shown that they look like a needle in a microscope. Then another type of phosphate stones are the struvite stones, and they look like a staghorn. Staghorn is something like a deer. Their horns and the struvite stones, they look almost same. So that is why they are known as a staghorn shape struvite stones. So these are looking very completely tracked. Because of their branches, they scratch and the bleeding is taking place. Crystals are mainly responsible for urinary stone problem. There are three broad categories: the calcium oxalate, calcium hydrogen phosphate dihydrate, ammonium magnesium phosphate, and all of these crystals are grown in laboratory using the gel growth method. So we are going to discuss later on about this gel growth method. Another issue for that crystals are very valuable, that is arthritis. It is due to the deposition of small crystals in soft tissues of joints. The crystals induce inflammation, particularly when they break. So arthritis is also because of the deposition of crystals. This is some, here some humor associated with arthritis. Cartoons are shown, particularly with the old people, and particularly the athletes have a knee joint problem. And sometimes they are advised to put chocolates, so lubrication is done very efficiently. The occurrence of arthropathies or arthritis. In India, prevalence of rheumatoid arthritis is 0.75%. In China and Indonesia, it is 0.4%. And arthritis affects 0.3 to 1% of population all over the world. And in world, in October 12th is celebrated as an Arthritis Day, World Arthritis. Now, crystals mainly responsible for arthritis problem. There are three main: sodium urate monohydrate. And all these crystals are responsible for the problems related to arthritis. First is gout. Gout is due to the deposition of monosodium urate monohydrate crystals in joints. Gout is also known as king of disease and disease of kings. And gout is mainly due to the highly nutritious food full of proteins. So in one of the journals of arthritis, the editor made a comment, it is this of kings and kings of disease. Why it is a disease of kings? Because in European countries in old time, people, the kings, they used to eat a lot of red meat and highly nutritious food. And because of the purine metabolism, the deposition of uric acid used to take place, and which used to induce gout. And why it is known as a disease of king of disease? Because it is so much painful, and that is why it is known as a king of disease. So it is a famous quoting that king of diseases and disease of kings. Now, pseudo gout is due to the hydroxyapatite, and the picture shows that we have grown hydroxyapatite small crystals in form of a ring, and these rings are known as a Lissagang rings, and the crystals are grown in the test tube in a gel medium. So popularly, the formula is CF5PO4 3OH, and the predominant mineral component of vertebrate bone and tooth enamel. So this is a very important biomineral. It is responsible for our bone strength. And whenever we smile, the tooth enamel is displayed, and white teeth is due to the component hydroxyapatite. It is the most stable and most compatible calcium phosphate form. And it is so stable that after the cremation, the asthiful is made up of hydroxyapatite. Pseudo-gout, formation of calcium pyrophosphate dihydrate, CPPD, crystals in soft tissues such as cartilage, meniscus, synovial tissue leads to the CPPD deposition disease. The appearance of these crystals in the synovial fluid can give rise to acute arthritic attack with pain and inflammation of joints, a condition called as a pseudo-gout. So the picture shows the cartoon related to the pseudo-gout, and one test tube, we can see that small crystals are being grown in the test tube. These are CPPD crystals. Now, atherosclerosis, when plaque or fatty deposition is taking place, it is called atherosclerosis. These deposits are made up of cholesterol, fatty substances, cellular waste product, and calcium and fibrin. So that is responsible for the atherosclerosis. Another biocrystallization is taking place in form of a cataract. Ferritin crystals in cataract. The calcium oxalate crystals are rarely found in lenses of eyes, and the unique structure of ferritin forms a spherical shell in which the iron is stored as Fe3 in a crystalline mineral. So the crystalline structure or the molecular structure is shown, and the crystals are responsible for cataract problem also. So the reference by D.G. Brooks, they have studied the ferritin crystals role in cataract. Then gold medal stones, these are hard stones and may develop if liver makes more cholesterol, and then the bile can dissolve it. So these are the particularly the gold medal stones or gold stones. The cholesterol, cholesterol is termed as a Jekyll and Hyde molecule, and we have a famous novel, Mr. Jekyll and Mr. Hyde. Usually two different faces are there. One is a very gentleman-like face, another is a Satan-like face. So the cholesterol is termed as a Jekyll and Hyde molecule. So it is useful as well as harmful also. A small amount of cholesterol is used by adrenal glands to form adrenocortical hormones by the ovaries to form progesterone, estrogen. And cholesterol and lipid substances control the rate of evaporation from the skin. So cholesterol is having a good effect, and they are also playing a role in a sex hormone. And it is mentioned in the literature that cholesterol is not present, the dehydration would take place very rapidly. So it controls the evaporation rate from our skin. And there are term as a good cholesterol and bad cholesterol. LDL that delivers cholesterol to our cells, and HDL brings it back from the cell. And that is why the LDL, low density lipoprotein and high density lipoprotein, they perform this work. And that is why they are termed as a good cholesterol or bad cholesterol. The human suffering in 2001, 17 million people died due to cardiovascular disease globally. In 1986, bypass surgery was 10% of the heart-related surgery in India. And today it's around 60%. And gallstone disease affects 12% people in USA. And this is just the introductory part. What is cholesterol? We all know about the cholesterol. Cholesterol is one of the most abundant and best known steroids in animal kingdom. It was first found in 1770 as a component of gallstone, and French lipid chemist Chauvel identified it in an animal fat in 1815. And the name is because of the cholesterol is from sterol, steroid alcohol, and that is originating the word cholesterol. The crystals are also found in plants. So I have taken an example. This plant is, it's a very popular, our indoor, and leaves are having small crystals and well-shaped crystals. The morphology is well-defined, and they grow within the leaf. Now, in vitro study, now we are focusing on gel growth, and these crystals are grown in laboratory by mimicking the atmosphere of human body by gel growth method. Now, here we have shown an example. The test tube is taken where the gel is set. So gel is having a gel structure. It's a porous structure, and it is a hydrogel. So the water is within the porous structure. And you are pouring a supernatant solution on the top of a gel. So first you have to mix a weak acid with sodium metasilicate, and that makes a silicate polymer, and that polymer is having a cross chain like a structure, and that forms a gel. It's a porous one. And in that structure, if you are putting a supernatant solution on a set gel, then that solution slowly diffuses into the gel, and the reaction is taking within the gel material, because gel is one reactant is already there in the gel, and another reactant is being diffused from the top. And whenever these two reactants, they react it with each other, the small nucleus takes place, and then deposition of nutrients takes place on that nucleus, and the crystal grows. The crystal growth is taking place in a gel medium. So the photograph shows a test tube where crystals are grown in a gel. They are not put in a gel, they are growing in a gel. Inhibition and dissolution study. Here the aim is to control or stop the growth of crystals which create human suffering. And ideally, crystals should be dissolved. So here we have to study the dissolution or inhibition of growth. Ideally, the crystals must be dissolved. But if it is not possible, then their growth is to be inhibited. And a very good book is written, that Advance in Crystal Growth and Inhibition Technologies by Z. Amjad, and that is a very popular book, and it's available in a market. technique is a growth inhibition and dissolution study. This technique is developed by one of my student, Dr. Bharat Parekh. Right now, he is in a Pandit Deen Dayal Energy University in Gandhinagar. And this technique is very simple technique. It is using a petri dish and 2 slides and cover slip. Usually in any pathological lab or any biological lab, these things are available. First thing is that you take a petri dish and you try to put a small drop of the gel solution. And once the gel solution is just thickened, then you put a cover slip on that. And in another slide, you put another slide on as a protective layer or as a cover, and then you put another solution in a petri dish, and that solution just diffuses into the gel, and small crystals are getting developed. And the development of crystal you can see under the microscope. If you have a digital camera facility attached to the microscope, you can record it and you can take photographs. So the method is described over here, and the photographs we have taken, that is on a brushite crystals, and the inhibition we have studied by using a lemon juice or a citric acid or citric acid. So the growth inhibition in vitro is carried out using the citric acid as an inhibiting solution, using a very rapid and simple method, because this is a very rapid method, very simple method. In a gel growth method, it takes almost 15 days crystals to grow. And if you want to study the growth and dissolution, again, it will take a few more days. So within 1 month, you can have a complete data that how much efficient the growth and dissolution is, growth inhibition or growth and dissolution is taking place. But here, the growth of crystal is taking place very rapidly under the microscopic condition, and using the inhibiting solution, whether the inhibition is occurring or not, you can check it very rapidly. Within 2-3 days, you can have a data with you. So this method is very important, and the paper was published in Current Science with Vaidya sir, and it was published in 2007. And the photograph of crystals appeared on the cover page of Current Science. Another is growth inhibition of calcium oxalate monohydrate crystals. And this is done by a very enthusiastic pharmacy faculty member from Pakistan, that is from Karachi. And he has adopted our technique, which I have reported or I have just discussed in the previous slide. He used that particular technique, and he studied growth inhibition in using the glass slides in a gel-based medium. And he used 3 different seeds or pulses, and the name of pulses are given. And these pulses he has used to study the growth inhibition of calcium oxalate crystals. And these pulses are very common. We use in our day-to-day life. So this technique is used by many people. Then how growth inhibition is occurring? In vitro studies, different herbal extracts of medicinal plants are used in a crystal growth experiments. In gel growth, these extracts are added on the top, and the growth rate of the crystals is observed. And aqueous extracts or alcoholic extracts are used. And these theories are discussed by S. Ahmed et al. And in his paper. So this technique is very, very efficient technique. Now I'm coming about the reverse pharmacology. That is the favorite subject of Dr. Ashok Vaidya. So I'm not a competent person to describe or discuss about that. But I will just observe few things about that. If the crystal length decreases, then it is called dissolution, which can be used for the treatment or cure the problem. If the crystal length increase becomes slow, then it is known as an inhibition, and that can be used for the treatment to give relief. This thing is very important from a reverse pharmacological point of view, because Sir has always advocated for reverse pharmacology approach. There are large varieties of herbal extracts available. Their toxicity, their potency, they are well documented. If we use those herbal extracts for pilot study in vitro, that can be very much helpful for paving a path for in vivo studies or animal studies. And we had one project with Vaidya sir and Mengi madam, and we completed nearly 15 years back. And that project, we worked in a tandem manner. We studied the in vitro thing using the gel-based model. And then sir studied in vivo and animal model things. And we came with a very good results in our DBT major research project. The herbal extracts, we used herbal extracts of Tuberous terrestris and Virginia regaluta, and these are used for inhibiting calcium oxalate monohydrate crystals. Again, as shown, the photograph is shown about the Tuberous terrestris and Virginia regaluta, and growth inhibition study was carried out by Dr. Mihir Joshi. Right now, he's a principal at Science College in Petlat, Gujarat, and he has grown the calcium oxalate monohydrate crystals in a test tube, and he used these herbal extracts to study the growth inhibition of these crystals. And the paper was published in Journal of Crystal Growth, and that is to be a very important paper. Growth inhibition of struve crystals. We used citrus indica lean juice, and significant growth inhibition was observed. And that proves citrus inhibition theory. So citrus inhibition theory, in a simple manner, one can say that if in urine the concentration of citric acid increases, there is a more probability of calcium ion to bind with the citric acid, forming a water-soluble calcium citrus salt. But if the amount of oxalic acid is increased in urine, then the probability increases of calcium to bind with oxalic acid or forming a calcium oxalate, which is a water insoluble one. So here the citric inhibition theory advocates that somehow if we increase the citric acid level in urine, there is a less probability of forming a calcium oxalate salt. And this paper was published by us. Dr. C.K. Chauhan, right now working as a faculty in a government science college, Gandhinagar. And we published this paper in 2008 in Urological Research. Then growth inhibition of cholesterol crystals. The cholesterol crystal growth inhibition studies was carried out by using Phagonia critica lin. And this herb is grown widely in Saurashtra University campus. So with help of bioscience department, we identified the plant, and from that plant, we got the extract using the facility available at bioscience department. And using this extract, we got a very encouraging result for inhibition of cholesterol crystals. The growth of cholesterol crystals is occurring in test tube in form of needles, and one can see in the test tube, the needles are grown. These are the cholesterol crystals. So the growth inhibition is occurring using the Phagonia critica lin. And this result was included in the PhD thesis of Dr. Purvesh Vyas. Right now, he's a faculty in Science College in Amreli. The role of tea in urinary stone. So we always prefer tea in morning and afternoon, and it is a common drink in our country. So tea is having a component which is very much helpful in urinary crystal inhibition study. So the effect of minus epicatechin, a component in green tea, is an precipitation and growth of infectious urinary stone amorphous precipitations. So this component, epicatechin, is an antioxidant and that is responsible for the amorphous urinary stone inhibition. And this study was carried out by a scientist from Poland, Joltana Priver, and she is working on growth inhibition of various crystals. And she is using a software. By using this particular software, she tries to predict that whether this inhibitor is a potent inhibitor or not. And usually for any chemical reaction, the potential energy should decrease. And that is the law of nature. So if two different ions or different active compounds, they form a complex, then if theoretically the potential energy reduces, then it is a potent inhibitor compound. So like that, she calculates the potential energy by using a software or computer program. And she predicts that particular inhibitor is a potent inhibitor. And that forms a good complex. And by that, she has selected several compounds. And in that, she has taken up the component of a tea. And that is used for studying the growth inhibition of urinary stone. Some of our research publications, the one publication is took place in Journal of Crystal Growth in 2005. The citation is the highest one, 105, and impact factor is 1.830, and of our group with the Vaidyasar's group. And the next publication is that is in a urological research, and that inhibition and growth of urinary calcium hydrogen phosphate dihydrate using the herbal extracts of Tribulus terrestris and Virginia ligaluta. And the citations are 76, and the impact factor is 1.45, and that was published in 2005. This is the cover page photograph of the crystals which we had grown using the glass slides and cover slips using the normal petri dish. And this is a photograph of a bruchite crystal, and that appeared on the cover page of Current Science on August 19, 2007. Another research paper, Journal of Material Science, Materials in Medicine, and that was published in 2009. And the citations are 85, and impact factor is 4.55. This is the round table discussion with Vaidya sir, another Vaidya sir, Vaidya madam, and myself. So this is a round table discussion. And we had a very hectic discussion on that day in Rajkot. This is a picture of our laboratory, the students working in the lab, and just myself looking into the test tube where the crystals are being grown, and students are discussing with one visitor. Our acknowledgements are, we are highly thankful to Dr. Vaidya sir and Dr. Rama madam. We had a very long collaboration with them, almost 25 years. We published high impact factor journal research papers with a citation over 100. And nearly 5 PhD students completed their doctoral thesis using our collaborative work. And one of my students, Dr. Mihir Joshi, he was the first person to work in this area. And a group of students, Dr. Bharat Parekh, Dr. Chetan Chauhan, Dr. Purvesh Vas, Dr. Kashmira Tank, and Dr. Sonal Vasan. So I'm very much thankful to my students as well as Vaidya sir and Rama Madam for their help and for their active guidance and all of and they produce good results for good publications. A concluding remark, crystal growth inhibition study by fruit juices and herbal extracts are very important for preclinical research and important for reverse pharmacology. This is forming important in vitro study. It is used by pharmacists for identifying potential herbal extract. This is multidisciplinary study. Thank you very much. Thank you very much. This is the photograph before my retirement in university office. Thank you very much.
Thank you very much, sir, for a very informative talk. It was excellent. It's a new topic that we could hear today, and it was very interesting. Ma'am, we could now take up discussion. So if anyone has any questions, you can please ask, sir.
Mihir bhai, excellent summary of many years' work.
Oh, thank you very much.
It appears so simple, but it is difficult to collate so many years' work and so many papers with high citations. And I think that we have left it halfway in terms of human usage. And that's why you rightly emphasized in the last slide that reverse pharmacology with these plants, with current tomography, other things can be done very well. And that's why you remember we had gone to the next door kidney hospital.
Yes.
I think we had gone there, but it did not work out. But I feel that our youngsters should again, particularly medical youngsters, should particularly explore this further. And I would say that in retirement, you're not retired, but we would rather want to have you as adjunct professor. And that is my request to our research director. And you and Ashwin can guide definitely this more, so it comes to light as a product also. But congratulations for a very good lecture. Wonderful.
Thank you very much. Thank you very much.
Yeah, Shobha, now it's open for discussion. I think
if there are any questions,
I find that still 19 participants. I'm pretty sure if we request everybody to put down their now video, see their faces, it will be a live discussion, I think. Share can be stopped. If the sharing stops, then we request both KIHS-MRF colleagues as well as colleagues from other collaborative institutes. Can we have that? If that goes away, the sharing, then we can have that.
sir, if you could stop the screen share.
I've stopped. I've stopped.
Yeah, okay. Thank you very much.
Hiteshi is there, Nirja is there, Dr. Meera is there, Sharvari is there. Anybody, please, if you have any question. I have some.
I saw Dr. Professor Joseph Mihir bhai. Is he still there? I would like him to comment at least first. And then any of your students who are now faculty members, if they are there.
I think I have given them the link, and I have put a WhatsApp group message that the talk will be at this particular time. But I think that they may be busy in their duties, particularly Tuesday is a working day, and people who are working in colleges, they may have some duties, so they may not have. That is why I suggested Shobha Iyer madam to put it in a Google Drive. So whenever they want to watch the lecture, they can watch at their own time.
Yeah, may I invite Dr. Hiteshi Dami? Hiteshi, can you put on your video also, please? How much is gallstone related to NAFLD? Are patients of NAFLD also likely to have more gallstones? Yeah, Hiteshi? I see your name here.
Yes, ma'am.
Yeah. So I would like to ask you a question and then I'll guide it to also the Mihir Bhai. Because as one of the things that we talked about when I talked about 4 mantras from a master, one of the things I said was that what are the things that are left, which Sir has already suggested. One thing is for atheroma, for sure. And that I'm going to discuss later on as Dr. Mihir Bhai becomes active after our invitation that is going to go as an adjunct professor. So that would be good. Now, the question that I'm asking Hiteshi you, are patients of NAFLD more likely to have gallstones? They do have cirrhosis and the cancer, but also gallstones are more likely with them?
It is prevalent in patients with NFLD. Not all of them, but yes, it is prevalent.
It is prevalent, yeah. Say, Mihir Bhai,
Yeah.
fatty liver is so common these days. Even with a milder BMI high, but the visceral obesity much more, and insulin resistance, they are the ones who have fatty liver. And then the fatty liver, it seems innocuous to many, is not so innocuous. It could be associated with gallstones and later on, of course, with cirrhosis and cancer. So I think that has picarosa curua been thought of for gallstones? Dr. Nutan Nabar is, I thought she was here. Has picarosa curua been thought of for gallstones, particularly for gallstones? Anybody from Ayurvedic is Dr. Ashish Bhatti there? Dr. Nutan was here a little while ago.
Yeah, I'm still here.
Yeah, tell me, has picarosa been advocated or being used in Ayurveda for gallstones?
Yes. Kutki is recommended. That is picarosa is
Okay. So, Mihir Bhai, as you very rightly
I
ended,
would like to add
yeah,
that picarosa curua is hydrocoleritic. Means whereby it increases the water content of the bile. And so there is something called cholestatic jaundice and cholestasis. And that stasis itself appears earlier in the biliary capillaries. But when larger things happen, they accumulate in the gallbladder, the stones kind of thing. And I feel that one area we had discussed, but we had not taken step was crystals of oxycholesterol, oxidized cholesterol.
Oxidized cholesterol. Okay.
Because
okay.
what damages in atheroma as well as everywhere is essentially oxidized LDL. And that oxidized LDL is the protein which is replaced. Not only so protein crystallization, we have some colleagues now who are working with us and they are very willing to work on protein crystallization with us. And I feel that we have to get Purvesh's paper out after he recovers from his fracture.
Yes.
But all his thesis is the only not published to my mind. All others have almost published everything.
Published, yes.
Yeah. So what I feel that picarosa is a very good example Rama has taken up. And what we need to do is some even in vitro studies about how picarosides 1 and 2, because Hiteshi has done her PhD on picroside 2 and picroiza on NAFLD, non-alcoholic fatty liver disease, in vitro work on liver cells. And but we have not studied bile acids, because I've been telling for a long time that there are bile acids which are derivatives of cholesterol. And so there is a cholic acid, there is a lithocholic acid. Now, lithocholic acid word itself means, litho means stone, as the urolithoacids. So the bile acid which leads to stone formation is more called lithocholic acid. And I think some of these things can be done in vitro after a good literature search and what others have done, as well as what is within our capacity to do.
May I request Dr. Shobha Iyer and Dr. Hiteshi, as well as Dr. Nutan, to send all our picoriza papers to Dr. Mihir Joshi, because I think we have opened up something which we have worked on for so many hepatoprotective plants, and our work on Picoriza curua dates back both in vitro, vivo, and in the clinic, Arogya Vardhini, and later on. So I think that Hiteshi, if you don't mind, since you will have that, the last JIM paper, if you don't mind, just send it to, if Shobha doesn't have, and then Shobha see that Dr. Mihir Joshi gets that. It's available in internet, but it becomes easier. So, and then we'll take it up that in one of our discussions on our Tuesday platform to take it further, because this is something which is very good. Dr. Pandey and others, VN Pandey, had shown what you talked about, that it becomes what you call it, hydrocholeric. Yeah, it's very interesting. Yeah. Dr. Meera, anybody else? Sharvari, Dr. Sharvari?
Yes, ma'am.
Any comment?
So it was really an interesting talk. It was a new topic, like very in-depth knowledge.
Yeah, for you it's new. For us, it is so many years.
Yes,
And
ma'am.
I wanted to get it to our platform, that
Yes.
one of the things that on Dr. Vaidya's birthday, 4 mantras, my first mantra was this, where Mihir Bhai appeared, and I have sent that slide to him also, that whatever we dig and which is covered, let's again uncover and start working on it.
Yes, definitely. Thank you so much.
Yeah. Shubhada, any comments from you, Shubhada? Sir talked about oxidized LDL. No, I think she's not there now.
Ma'am, I have a question for Dr. Joshi.
Yes.
Question is it possible to isolate these biocrystals intact in the laboratory from human fluids without disturbing the crystal structure? First thing.
People have isolated them, and they've studied extensively, and particularly the urinary crystals, they have done it. Hydroxyapatite, they have done it, and cholesterol, partly they have done it, because usually they are making a complex with the other compounds, so it is slightly difficult. But mineral crystals are very easy to isolate, very easy to isolate. And the first people isolated them, they took the powder exit diffraction study for identification, and they identified them as a particular mineral crystals. And then people started working on the reverse manner, trying to grow the crystals in the laboratory, which factors are affecting the growth, and then inhibition and other studies. So that is what we are focusing.
And my next question follows this. Your lab-made crystals, which you showed in gels, do they truly mimic the biocrystals?
Yeah, in a certain aspect, they try to mimic the biocrystals. We know that in human being, the things are very complex. And in a soft tissue, the crystals are being grown by supplying the nutrients from our body fluid itself. But in a laboratory, we have to mimic that situation by creating an environment of a soft gel. That is something like mimicking the tissue. And it is a porous one, so that the fluid is being transported, being supplied, and that nutrient or fluid is having a composition of the different nutrients, so that the usually the compounds are being supplied to the site of growth and the crystals grow. So it is almost a mimicking, not making the exact atmosphere. That is why the term is used, mimicking. It's not exactly the body condition is made. So body condition is always very complex, but we try to make it in such a way that we try to understand that how they grow.
Can we have the name of the person? Can you show your face and who's asking this question? We would love to see you.
Yes, ma'am. I will do that.
Yeah, lovely.
Am I visible?
Oh, Dr. Meera
Yes.
was asking. Wow, very nice. Oh, madam.
Yeah.
I thought as much though.
Sir, my next question is this. Since you say you mimic the natural crystals, I just wanted to know, because the osmotic conditions that you would be maintaining in your gel would not exactly be the way that it would be in, say, within a human system. So when you say you're using it to reverse crystal formation, is that going to impact?
Yeah, it's a good
same
question. Good question, madam. That was when we started working on this system long back, because it's a big story. I met Dr. Vaidya nearly 25 years back in one of the conferences in Rajkot. And since we are from the same family, so we know each other. And I asked him at that time casually, are you working on kidney stone? He said, exactly not working on kidney stone, but I worked and I have a good expertise in kidney stone. Long back, I used to remember that he worked sometimes on kidney stones. Then I told him that one of my students is also working on the kidney stone. And then we started collaboration. And that collaboration started with his knowledge of Ayurveda, his knowledge of clinical pharmacology, and our knowledge on physics, as well as characterization and growing crystals. And in a first lot, we did a very simple study, and that work is being published. And that work is published long back, means almost 15 years back. That is very simple work that we use different herbal extracts. But your question is very important, that how to mimic the condition. In kidney, it's a dynamic one. It's not a static one. The urine is being continuously generated, flows through the system. So we have to grow the crystals in a dynamic system. Another thing is that what are the conditions within the urinary tract or kidney? pH level, as you mentioned, osmotic condition. So again, we have to take a help of medicine or pharmacology people and try to create the atmosphere, the dynamic atmosphere. And we try to see that how crystals grow in vitro, not in a system. And we try to use the inhibitors, that how they try to inhibit it. Our system right now, we study it, it's a static one. But we want to have a dynamic system, as you mentioned, that using different conditions, pH level, then osmotic condition, all these factors we have to consider. Again, we try to repeat the thing. It will be much more nearer to the real situation. Not exactly the real situation, but nearer to real situation. So
Doctor, may I come in, Meera madam?
Yes,
Reverse pharmacology starts with clinical experiential data or exploratory data or case reports. So the plants we have chosen are used for centuries in kidney stone. So when we want to test it and we found it positive, it is already in human positive.
Oh,
And so defining conditions may be different. For example, as Mihir said, in terms of citric acid, citrate containing fruits, all that. And but I would like him to say something of your interest. That is how microbes may play a role in crystal formation. Mihir, please, because she's a microbiologist and she would be very interested there.
Accidentally, we found one interesting result, but somehow we could not work on that because of our less expertise. We were growing crystals of potassium tartrate and strontium tartrate in a gel beater. And we found the gel becomes dirty and some cotton-like thing getting attached to the crystals, and it stops growth of crystals. Then one fine day, I invited one of my colleague from bioscience department. She used to be a very good friend of mine. So I called her and said something dirty is taking place, some cotton-like things, threads or something like that, because we were not expert in that particular field. She immediately identified these are fungi. And these fungi are attaching with the crystals. It stops the growth of crystals. Then she identified the fungi and we published one paper by doing characterization of that. Later on, one of my students was working on a modern sodium urate monohydrate, and he experienced the same thing. And in at one this monsoon atmosphere, some fungi attack took place, and it stopped the growth. But it did not happen again. So it was accidental or whatever maybe. Then we showed the result to sir, and the sir was very much keen, but it did not take place somehow because it was accidental one. So we were not expert to identify. And sir said that is a very important result. These fungi may be working as an inhibitor or maybe having some bonding on the crystal, some bonding on the crystalline surface, because now the molecular docking is a very important branch of chemistry. In that one, the active site of fungi as well as the crystalline molecule, they are being docked, and the potential energy, everything is being counted, and they try to do the modeling whether this site binding on the crystal and will inhibit the growth of crystal or not. So that is a theoretical one, and they are using different type of computer software, and they predict that this fungi or this particular microorganism will affect positively.
Yes, sir. That actually the reason that led me to this question was that if you've got just like opioids, you can have compounds from fungi, which probably can be used as pharmacological substances, which can be consumed if they are not toxic.
Yes,
you can
yes.
just consume them, and then probably they may act as inhibitors for, you need a foci for the crystal to form. If you can just stop the foci from forming itself, then there's no question of crystal formation itself. I mean, I'm just throwing a wild idea, but I really don't know how much it can be translated. Maybe
Madam, your idea is absolutely important, and that we discussed with sir, and we found a similar type of result, but unfortunately, it was not observed again. That was an accidental one.
Okay.
Meera Madam's question has raised some thoughts in my mind, that when you have grown crystals, which we could see it in the test tube, and you showed the urine also with oxalate crystals. If you were to separate out those crystals from the test tube, would it have the same morphological features as that you see in the urine?
May not be same, because the conditions are different. Even for the kidney stones grown in different patients, some they are having a very rough surface. Some are having a staghorn type of thing. Some are having a very smooth one. So different conditions, pH, temperature, then the nutrients being supplied, all complex things are taking place, and that is why the morphology changes.
So
But
one more question on that only. Then how do you characterize and reproducibility for that particular set of experiments?
but we are using the nutrients which are pure material supplied by chemist.
Right. That
So
is not
we are
growing.
sure that
Yeah, culture material is there. which
it's
is
not
constant.
culture one. So culture material we are not using. So these are the pure material supplied by chemist, means a chemical supplier, and we are using that. So for example, calcium oxalate. So we use calcium hydroxide and oxalic acid. So by just reacting calcium hydroxide with oxalic acid, we get calcium oxalate. So it is a pure one.
Sir, instead, what if you were to isolate? I mean, you said it's possible to isolate these crystals from urine or from whatever body fluid. What if you would do the same experiment using patients' crystals? Would it work?
It's a very good idea. But madam, I told you that different pathological conditions, physiological conditions, the crystals obtained in urine, they have a different shape and different particular conditions. So first we should have a patient history, under which condition these are grown in a urinary tract. And when we recover, then what was the condition of a urine? And on basis of that, we can isolate them. It may not be possible that the crystals isolated from one patient is having a same morphology, same condition from the patient recovered from other patient. So we have to first take the history of the patient as well as from the data collected. And then we try to do that growth inhibition study. Then we can identify whether it is occurring or not. It's a good thing.
Thank you, sir. Thank you.
Usually, madam, the crystals recovered from urine, they are having other components also. For example, uric acid, some proteins associated with our body, and some organic deposition also associated with the crystal. So it's a not pure crystal. And usually people have done the elemental analysis of crystals, and we have published one paper also by doing the analysis of the kidney stone, not crystal, the kidney stone recovered from a patient. And that gives that it is not a single phase compound. It's not a pure calcium oxalate, but it is having an impurity of other compounds also. And that impurity may play important role.
So hydroxyapatite, which you were mentioning, is routinely used in many protein purification. Protein chemists routinely use this because you can generate hydroxyapatite in a laboratory with a very simple chemical reaction.
Absolutely right, madam.
But what happens is hydroxyapatite, depending upon the cross-linking, can act like a gel or a sieve also. It has some sieving capacity also.
Yes.
So
it is used, hydroxyapatite is used for the targeted drug delivery, for separation of DNA, and for many biological applications it is used, and it is a very important biomineral.
Right.
Madam, in fact, I have a set of the presentation where I discuss about the growth of hydroxyapatite in the laboratory. And we made a nanocomposite of hydroxyapatite. And as Shobha madam mentioned in the introduction, we made a composite of hydroxyapatite with curcumin. And we know that curcumin is having antibacterial, antiseptic, and many other biological activity. So we combine that with the hydroxyapatite, because hydroxyapatite used as a bone ceramic material. So if you are using hydroxyapatite with curcumin, then you are having extra benefit.
May I come in?
Thank you, sir.
I think that this problem of microbes, bacteria, fungi in stones, particularly calcium oxalate, brucite, struite, urolithiasis, there is a paper in 2021. And earlier 2 years also there are papers. And actually, sometimes it is believed that you need the nidus of a microbe to truly have a reposition around it. And I think this paper I will request Shobhaya to send to you. And it is in Kidney, a very reputed journal. And it is Kidney, Kidney 2098 to 298 to 311, 2021, Kidney, volume 360 and 2. So if we get this, or even if you just put search as this, it has also given many references. It's an extensive review and a very good study also. They have also identified the microbes, the wonderful photography. I don't know whether you can see this or not.
Oh, yes. Beautiful photographs.
So it is shown that so there is an interest continuing in terms of and if you see here, there is a microbe which is actually inside the stone.
Yes.
And nearby recently it has been found that microbes are in many tissues. Up till now we felt that microbes only symbiotic microbes exist in lungs and now mouth, nose, skin and GI tract. But now they are finding in many organs actually by microbacteria, which even sometimes the species identification has not been done also. Same thing applies in this paper I referred to. So it is very essential to, if we want to review again, what is the current state? Because it is well known that those with urinary infection, they are more prone to stone formations. So I would say that the plants which we have studied and found very good effect, we have to continue further, and we may also take help of Meera Madam for microbiology part of it. Thank you.
And these two white crystals are because of the urea splitting organisms, and they are responsible for the certain microbes are responsible for the true white crystals growth.
Great. So, Mirba, it was an excellent talk, and you
Thank you very much.
generated a lot of discussion too.
I have one more talk that is on the much more technical one.
So
I have not.
we'll have a second talk in a due time.
But in next month we'll try to give.
Yeah.
Yes.
Yeah.
Because that will be much more technical. I have not given any data right now. This was an introductory one.
Yeah.
So I have not given any data in this particular talk.
So
And I have one talk about the nanohydroxyapatite and its applications. So Madam suggested that nanohydroxyapatite is very important one for many biological applications.
Thank you very much, sir.
Thank you very much.
Thank you.
Thanks.
Thank you very much.
Thank you, sir.
Thank you.
Yeah, very nice. Lovely.
Thank you.
And since it will be recorded and it will be available.
Yes.
Right, right. send it across to Dr. Mihir Bhai, of course. And then I don't know where Rhoda will know about it. Are we putting in our website or we are putting it, where do we?
No, actually, it's a very huge file. So I will be sending it to everyone personally, ma'am, so that everyone can have an access to it. Since it's a huge file, we will not be able to upload it on the website.
So, Shobha, we can upload it. We have a YouTube site also.
YouTube site.
So we can upload it there and we can send the
Yeah,
link.
then we should
sir.
keep it on YouTube.
We'll
Yes.
send the link to sir.
Yes, that would
Thanks,
be.
Rhoda.
Yes.
I will send the original link also. Also, this YouTube, once we upload it, we can send it to you, sir. That
You
also.
can have a 2 version. If you want to upload only talk and talk plus discussion, whatever is possible, then you can edit it and you can upload it on YouTube channel. So that will be giving a viewership on a larger scale, and
Yes.
people can have a view of that.
Sure.
Thanks.
Thank you very much.
Siddesh is there.
Yes, ma'am.
I will do this with Siddesh maybe tomorrow or something. Yeah. Okay.
Sure. Thanks, Mihir Bhai. Thank you.
Thank you very much. Thank you very much, Ramaben. Thank you.
Shama, thanks, and thanks, everybody.
Pranam Ashok Bhai. Yes.
Yeah, thanks.
So I leave the meeting. Okay. Thank you.